The next one up is Stephan Brock, Molecular Health, Engineering the Medical Revolution, Big Data and Clinical Decision Making.
Okay, thank you very much for the invitation. A great pleasure to be here. Actually, I couldn't have wished for a better speaker slot here. Garrett was extensively talking about all the challenges that we have in data generation and all the nitty-gritty stuff that we have to observe. Werner was talking about technology for real-time data analytics. So I can focus now on the big data thing. And actually, I split it in two pieces. I split it into one piece being the application of prior knowledge. And we've heard that in Garrett's talk. I mean, he was talking about, in the end, what really matters when we look, for example, at the interpretation of genomic data, it's the evidence. And this is a moving target. I mean, we have a ton of new publications every day. We need to keep up with the development. We need to provide context. We've also just heard a question here from the audience, what about data quality? It's also something that actually has to do with this application of prior knowledge. We can use such methodologies actually to clean up such data and to actually reference it to one common annotation system. So for me, the application of prior knowledge, it's a kind of outside-in perspective. So we're looking at one patient and we want to contextualize that information that we get from the patient. So it's a molecular profile, it's a clinical profile. We want to contextualize that. So we need to structure all the knowledge that is out there. Then the second piece I'm going to address is actually leveraging real-world evidence. So what does that mean? We're treating patients every day, we're collecting outcomes. Increasingly, we're actually assessing molecular profiles of these patients. That's all a great database to learn from. So we can actually learn from the accumulated data and we can inform future decision making and the principles how we do that and how we address that I'm going to actually talk about in the second piece of my talk. So first one, application of prior knowledge. This is something that has to do with very much the classical bioinformatics theme about data integration and data structuring. And we have all these different data domains, tons of data out there. When we're talking about big data, actually it's not so big data, but still it's a lot of data that you need to integrate here. And one of the key challenges that we have here and that we're facing is, even today in the age of IT technology, how is new knowledge stored? Not in databases, it's published. We have our publications, all great results, all clinical trials, it's published. Not in databases, it's published in journals. So this is one challenge. How do we get that into a database so it becomes computable and usable so we can use it for the interpretation of clinical molecular data? I think last year I talked about this application for clinical decision support here, and my example for data integration was the data around the drugs. So today I'm going to use another example. Just one thing I should mention, we're just branding that newly. Our marketing department decided that our integrated knowledge database that should be called Ingenious because it's the engine that drives everything. So let's do them justice. I mentioned it here. So one example, clinical trials data, typical journal article about clinical trials. So I'm now talking a bit about the engineering approach. How do we get the data from the publication into our database? So we're using text mining, and text mining actually extracts like the makeup of the trial. It tries to connect it to the clinicaltrials.gov database, and it extracts clinical metrics. So actually this LUX3 trial, it's exactly that publication. That's what it looks like in our curation. It's an internal interface that we're using. So data is automatically fed into that. Also the kind of biomarkers that are assessed here. this data is then assessed. I mean, we know what the cohort size is, the age distribution, the ethnicities, and so on and so forth. The biomarkers that have been assessed, and this is then subdivided into the treatment arms. And for each treatment arm, we are actually recording the outcomes metrics. So we really get down to the outcomes metrics. And in the end, when we have all that data, we can do arm-to-arm comparisons, calculate hazard ratios. Once we have that for a number of trials, we can also construct in silico kind of new trials from that data. So that is a nice resource by itself. So we're not doing that for fun, we're doing that for clinical application. We're trying to keep all this data up to date. So we're using it in this test scenario for clinical decision making, but not only for clinical decision making, we're using that data also for informing the design of our tests. When we do panels, we actually prioritize the genes that are going onto the panel with our IT methods. Then we're actually looking along this process for the actionable biomarkers. So we're matching that against our database where we have all this knowledge present. And we're also using that for the huge number of variants of unknown significance. I mean, once you have such a database, you can also do inference. You can do structure-functional inference here to actually help the interpretation much better than the statistical tools for the interpretation of variants of unknown significance. The most important is the clinical evidence grading that we do in the end. So this is a process that we've implemented, and we're supporting this process and will support parts and pieces of this process. So we really have a platform here. We also support with our software clinical labs, hospitals, IVD providers, and so on. There will be announcements soon about actually the release of that platform product. Just to quickly go through that, presented that last year already, how we're using web interfaces to actually present that data, so where we're ranking effective, ineffective, or toxic therapies, give all the background information on the variants and biomarkers that have been identified, also link that actually to different evidence levels, to publications, so you directly have all the information at your fingertip, the clinical trials, also the drug information and background information, detected biomarker, non-detected biomarkers, and safety biomarkers, and so on. And if we have the data available, we also contextualize that within the patient's timeline. And in the end, we have a report generator that is actually used to generate such a report, and that's focusing on treatments that work, prioritized by evidence, so the approved drugs, off-label drugs, drugs in development, drugs that are potentially ineffective by the nature of biomarkers, and also drugs that come with increased toxicities, adverse events. That's where, for example, germline signals become very important. Just one example, I mean, we had that structuring of the trial with afatinib, with the exon 19 deletion, so that's actually a nice example to show here. That was a pretty old elderly lady here, which was actually the mother of one of the key opinion leaders that we're working with. And she was treated for her non-small cell lung cancer, and actually was a candidate for hospice care because she didn't respond to Tarceva anymore. But her son decided to do actually the test, and we identified, yes, lack of efficacy for erlotinib/Tarceva, but actually afatinib should work, and that's one of the many examples. If nothing but that, it significantly and immediately improved the quality of life of that patient. Just to highlight the importance of actually germline information, just want to use the first example here, the well-known BRAF V600E. So you could actually use dabrafenib or verumafenib, but at the same time we detect actually a safety biomarker that's also part of the label for dabrafenib and indicates hemolytic disorders, a high risk for that. So here this information can actually be contextualized and used to choose verumafenib over dabrafenib in that case. I won't go into the other cases. So that's how we're actually employing this outside-in approach, actually all this aggregated evidence. So let's go to this inside out. When we collect a lot of patient data, a lot of outcomes data, we can actually build and generate novel insights. So new insights from patients' outcomes. First examples I use is we took the FDA's adverse events data, so safety data. These are very fragmented patient reports. It talks about the individual patient, his indication, so the disease, the drugs that the patient has received, the outcomes and the side effects. Pretty dirty data, so first thing is you need to clean that up. Once we clean that up, we can use the first approach, which is our knowledge model, to enrich this data. So actually, here we have more than 7 million patient records, and now we add to that actually all the mode of action, the targets, metabolizing enzymes, the drug classification, pathway information, and so on. So we add value to that data. So that gives us a new perspective on the data, and we can actually exploit that data. So here are just some numbers that we have derived then from this integration and cleaning approach. So we can do a ton of statistics. Actually, the FDA recently licensed that technology, and in the meantime actually has already led to 5 label changes for drugs that are on the market. So that's quite remarkable. Just want to give you one quick example. So here we're looking actually at one patient who received erlotinib. So this some basic demographics, the outcomes, the drugs, so quite a number of drugs, and all the multiple reactions. And actually a different representation that we can do for this data now is actually, these are all the drugs that this patient has received. These are the targets, metabolizing enzymes. So you can actually search through that data, scroll through that data. And one of the use cases that we have here, here I've highlighted one cytochrome, and this cytochrome gets a heavy overload by the polymedication here. This can be one reason for the side effects. Actually, we're using that technology for patient de-risking when we're looking at polymedication profiles. Just wanted to give you one example how we can exploit this data. So we're looking at a cancer drug, erlotinib. We're looking at all the side effects, and now we can actually screen for individual organs. So here I selected skin. Dermatitis, acneiform dermatitis is actually one of the signs that the drug actually works in the patient, which is pretty interesting. Actually what we can do when we have all that data in one system and everything is connected, we can zoom in on acneiform dermatitis, and it gives us all the drugs that are interacting with acneiform dermatitis. So that's a new concept that we're looking at. And now we're looking at molecular mechanisms, and when we look at molecular mechanisms, not surprisingly, top one hit is keratinocyte differentiation. So now we can look at what are all the drug targets in keratinocyte differentiation. We can look at the drug classes involved, and second to the anti-neoplastic agents is interestingly immunosuppressants. Yeah, because TNF is part of the pathway. But now we can actually look at the immunosuppressants like infliximab, and now look again at skin-related side effects. What we see interestingly here, we have skin cancer, basal cell carcinoma, malignant melanoma as side effects here. And it's this specific cancer type, it's not across cancer. So what we have here is actually a phenotypic expression of the reciprocal activity of infliximab versus erlotinib on keratinocyte differentiation. So in other words, it gives you ways to model the data, work with the data, generate new hypotheses on the fly. So that's pretty interesting what you can do. Second example for actually how we're using patient outcomes. This is actually a figure that I have taken from a Nature publication where actually a group worked on the TCGA cohort for endometrial adenocarcinoma. And endometrial adenocarcinoma, classically they are looking at the histology of that disease. So serous versus endometrial. Serous is high risk, so patients get adjuvant chemo, and those who are endometrial at low risk, they don't get adjuvant chemotherapy. And we know that in about 25%, it differs from where you look at in the literature, we have a false classification. So either significant under or over treatment, which is both bad for the patient. So what the researchers did in that case is a complete genomic profile of that patient cohort with patients with endometrial adenocarcinoma. And they used all the TCGA data, multi-platform data, to generate novel classifiers to get a better risk stratification for these patients. So these are the Kaplan-Meiers for the 4 new subgroups which they built. So they looked at the mutation data. They created one group which has mutations in polymerase E hotspots and is ultra-mutated. Then they have the microsatellite instability phenotype as a second class. And all the remainders have been grouped according to transcriptomics data in high and low expression clusters. So it's a pretty complex, a very complex profile that they came up with. And actually we took this data as a test case in our collaboration with SAP. And I'm taking a live look into that data now, so I hope the network works. So fortunately, this is not our server. If it doesn't work, Werner is to blame. Okay, so what we have here is 248. It's a small cohort, but it works similarly for large cohorts. I mean, that's what the technology gives. So we have the 248 patients here, and we can actually look at the Kaplan-Meiers and at the censoring events. So that's the background cohort. Now what we can look at, how is it done in a classical manner? So actually what we can do is add the histology. So we have the low-risk profile, the endometrioid histology, and we have the serous histology, which is the high-risk profile, very nicely represented in that data. We can also actually remodel the complex classifier, which they generated in that Nature publication. And this is called integrative cluster here. So we can very nicely reproduce the Kaplan-Meier curves here for this novel classification. So now when we look at that, we see polymerase E, microsatellite instability, all has to do with DNA repair defects. So we took a very simple approach and said, okay, let's approach it from a very simplistic way. We look at what are effects of defects in DNA repair. It's a high mutation rate and it's increased transversion rate. So we created very simple metrics here, and actually we can go to mutation rate. Mutation rate alone gives a pretty nice separator here. And now when we add the second hallmark of DNA repair defects, which is CA transversion, actually we come to a situation, we have a very similar separation with very simple metrics, and when we look at the statistical power, it's pretty comparable with what we have with this rather complex methodology. And very interestingly, the 17 patients that are in the lowest risk group here, in the blue curve up there, are identical to the lowest risk group of this really complex classifier. So that took just 15 minutes working with the data to come to this conclusion. But what we can do now is actually look, how does that actually look when we superimpose it on this original integrative cluster. And what we see is a split up especially of the microsatellite instability group. So we can actually zoom in on this MSI group and look at what the impact is. And actually, when we just look at simple metrics such as mutation frequency and CA transversion, we can split the previously homogeneous group of microsatellite instability into 2 subgroups here. We also took a look at the potential implication of some genes, and of course, when you think about such a cancer type, endometrial adenocarcinoma, you want to look at the impact of hormone receptors. So we simply looked at estrogen receptor. An estrogen receptor is very susceptible from a functional point of view to any kind of mutations. It's a very sensitive protein. So We simply looked at whether the patient has mutations in estrogen receptor or not, and this is the result what we get. It's not significant in numbers, but it's a very interesting trend because the orange curve, these are the patients that have at least one mutation in the estrogen receptor. So that could spur a new hypothesis. So you could think about, well, is that cancer type or this subclass of the cancer type estrogen dependent? Is there estrogen addiction, perhaps? Something to actually investigate further. But I only want to show you that actually this way of dealing with the data in real time, constructing cohorts, looking at the effect, having a direct impact on the outcomes, actually spurs the ideas on the fly. And the greater your data set is, the more hypotheses you can generate. And you can also use that data and split it up for in silico validation and then take it further from there. One last point to this. Just imagine, and this is something that we are currently working on. I'm doing a thought experiment now. You have a patient with a clinical molecular profile. So you know the clinical profile, molecular profile. You have a huge database of patients where you have recorded similar data. Now you do patient matching. You say, okay, give me patients with a similar molecular profile or a concept match like druggable pathways affected or things like that, and a similar clinical profile, and show me the outcomes of the different therapies that these patients underwent. So imagine you have that and you get these plots, and this one is therapy A, B, C, and D, and you can immediately see for a patient in the same situation what is the best treatment. So this is kind of the next step when we're talking about really big data and the application of that big data in a clinical setting. Talking a little bit in the future, but actually this is something that we currently have under development. Okay, this was my backup. Last thing, I cannot possibly skip that because it's such exciting news. We got on the cover page of Cancer Cell for just this current issue, which actually came out on November 9th. And I was already wondering how the editors would actually transform our story into the title page. So they actually chose to go for a cartoon. So let me explain you the story by explaining the cartoon. So what we have here, the background is actually chemotherapy-induced anemia and the treatment of that with human recombinant EPO. So this is the therapeutics that come. This is a human recombinant EPO. These are the guys, these blue guys are the EPO receptors. They take the EPO and they actually start the engine to produce red blood cells. That's what we want to have. But what they identified in cancer is that actually if you give EPO to a cancer patient in certain cancer types, it actually furthers tumor growth. And these are the red guys. So the red guys, they are ready to accept EPO as well. And they trigger an engine that actually furthers tumor growth. I think that nicely explains the principle. So coming back to big data and what we're doing here and how we have applied it, we actually said, well, this tumor growth mechanism must be different from erythropoiesis. So we came up with a number of questions. We had our integrated database, so we turned these questions into queries to the database. We answered all these questions here around structure, regulation, and function that would be actually conducive to this novel EPO receptor. And then we scored the proteome, and actually the scoring mechanism is pretty much the same scoring mechanism that today we're using for prioritizing treatments when we're doing treatment prioritization in our treatment map. So that was a starting point. It took us 2 weeks to come up with the hypothesis, and then the validation. That was a long term, but here we also took this outside-in perspective. We looked at patient cohorts, patients who actually, where we have clinical outcomes, where we have treatment information, where we know whether they had been given EPO or not. So again, the cohort perspective on that. And our novel EPO receptor, the biomarker, actually is known as EPHB4. When we look at the biomarker negative population, basically doesn't make a difference whether they get EPO or not. When we have the biomarker positive ones, first thing we see, it's a kind of a prognostic overall biomarker, but it's also predictive when we talk about the use of EPO. And the difference in median survival between patient who had no EPO and patient with EPO, this is ovarian cancer here, by the way, difference in median survival is 2.5 years. So this is a really, really strong biomarker. Now we have finally also the clinical proof. Further validations are ongoing. So actually, these are 3 examples. The one with the AERS data, pure clinical data, using this outside-in approach, we can enrich that data. Using all this rich and large numbers of data, we can generate novel hypotheses. The second one, how can we use clinical molecular profiles and outcomes data actually to generate novel hypotheses and test these hypotheses, and in the end also use that for informing treatment decisions. And the last one, actually, just for the news of it, because it's such a nice and exciting story. And I believe, especially when we talk about the outcomes data, this is really our challenge. There's a ton of outcomes data available, but here we truly have a bottleneck. How do we get it out? This is also our challenge, and this is a challenge that we truly need to address jointly. So that's the end of my presentation. Thank you very much for your attention.
Thank you, Stephan, for an exciting talk. Questions? There's one up there.
I wonder if you can predict advantageous but perhaps non-intuitive drug combinations. For example, there have been advantageous effects due to aspirin use, which might have a pro-apoptotic effect and can be effective in combination with a cancer treatment.
Yes, we have quite a number of these examples. Just let me quote one. Actually, we once looked at this patient safety information, at all the safety data, and looked across all cancer indications and all different cancer treatments. Is there any co-medication that reduces overall mortality? Because we have death rates reported. And interestingly, it turned out that across the different indications and across all different therapeutic regimen, it was beta blockers. And we also tested this hypothesis whether these patients had kind of cardiovascular comorbidities, or they took actually medications that were inducing cardiac failure or whatsoever. So we ruled everything out. And actually, that's also some work that has been published in Nature Communications with our collaborator from MD Anderson. We also identified the mechanism. So we can actually, to answer your question, we can exploit and use that data also to identify such beneficial co-medications.
I have a question regarding the text mining. So I understand that this is one of the very important topic in computational biology at this moment, yeah. But it's difficult as well because of non-existence of any common standard in the texts, at least the old manuscripts. So there has been some, currently we can see some community projects going on, like community curations. For example, the EBI and NCBI, they are taking lots of interest, I mean, both for automated curation as well as the manual reading of papers and kind of validating whatever they are entering into the databases, for example, the reaction database. So my question is, and also I really want to thank you for your initiative in this regard, but my question is, like, are you also considering those community curated resources, or you are curating your resources by yourself? My first question. Second is, like, if you are curating by yourself, whether you are only relying on the automated semantic technology, or also you are relying on manual curations. And if you are doing it by yourself, then what is the overlapping statistics with the community curation resources at this moment?
The very simple answer is it depends. So Also, the major feature upon which our strategy depends is actually the required quality level of data that we need to embrace. So when we think about the highest quality requirement that we have is really around the clinical data that is actually used for treatment decision making. This is something where we own the complete quality management and quality approval process. So in the end, we don't care about where that data comes from, whether from external sources, whether from external curation efforts or internal curation efforts. Whenever we need this highest level of data quality, we do the quality assessment ourselves. Now, when it comes to the availability of data that comes from these public efforts, this is something we are ready to embrace. So we are constantly scanning what's going on there, also what kind of technologies are used. We are relying on the competitions that are out there. As you know, there are these competition events testing the extraction of different facts from the literature, where they're always looking at precision recall metrics. And we're not reinventing the wheel. So if there is a good technology out there, we try to license the technology or implement technology. If nothing is available, we tend to generate that ourselves. Now, we have cumulatively about 15 to 18 years of text mining experience, not only from this company. I mean, I worked with some of my co-workers in other companies before, so there's a ton of expertise here. I mean, this is really one of our key assets in the company. Okay, thank you.
Any further questions? That's not the case. Thank you very much, Stephan.